So we have a couple of great researchers joining us today, and we're going to kick things off with a talk by Peng Jiang, who is an assistant professor who works at the Center for Gene Regulation in Health and Disease located at Cleveland State University. And Peng's research is centered on developing computational methods to leverage massive genomic and transcriptomic data to investigate the temporal, spatial variation and dynamics of gene regulation. And so give me just 1 minute to switch up our screen share here. And Peng, whenever you're ready, feel free to share your screen.
Yeah, thank you for introduction. I will share my screen. Can you see my screen, the full screen?
Give me 1 moment to move the screens around.
Yeah. So is that full screen?
Okay, perfect. Yep, you're all good.
Yeah, okay, thank you for introduction. So today I'm talking about computational methods for comparative, I'll move the screen a little bit. Yeah, comparative time clock in early development and tissue regeneration. So basically compared to other speakers, I'm adding more biological component for this section. Okay, so so we know the biological clock are internal mechanisms that control the timing of biological events such as circadian rhythm, development, and aging. So we know that circadian rhythm usually manage the pattern over a 24-hour period of time. And for development and aging clock make us from the young to the old. Every clock functions with it is a distinct timing and pace. Let's say, for example, for the development clock, the gestation period for the mouse is significantly shorter than human. Let's say the mouse will take only 21 days for the gestation period, but human, we know, will have 38 weeks. So the dramatically differences in the timing. So basically, every clock function is distinguished timing and pace. The pace are completely different. And the biological response to a clock is complicated as well. Let's think about it. We have circadian rhythm, and let's see, for tissue regeneration in terms of wound healing, the time clock in wound healing actually is unevenly distributed. So this is just an example. Let's say this during the day. So basically during the day and during the night, actually the healing in terms of cell activity completely different. So basically during the day it's more quicker, but at night it's very slower. So there are many differences. Basically the biological response to the circadian rhythm or to the clock are dramatically different and complicated. And the clock itself actually can be complicated as well. Let's say the internal clock is uneven. For example, let's say the early development for the frog or for axolotl. During the axolotl embryonic development, there are fluctuations between the transcriptomic disruption and stability. So specifically, when we think about the early development, we're always thinking about it's even. Actually, it's not. Let's say from the embryo, and then started to differentiation and go through all the justification period. And what we are doing is we are looking at each hour, then calculate the number of the y-axis is the number of differentially expressed genes, and the x is the time point. So basically, during the early development for the axolotl, there are certain timing have dramatically transcriptomic changing. So basically, for internally, the clock is not even. They have some up and down. So basically, they have the transcriptomic burst and stable later on. They're complicated. So what we have been known is the development clock in the mice is much faster than human. This is based on the morphology. We can say it based on morphology. And we know the embryonic stem cell differentiation speed in the mouse is faster than human. It's based on the cell level, we can say it. And for the tissue regeneration, we know axolotl can fully regenerate the limb because of a cell called blastema. Basically, the blastema can replace the lost link after the differentiation. However, the mouse can only regenerate the partial of the digit. Specific means digit tips. And we human have almost no regenerative capabilities. We cannot regenerate link. So the question is, we are talking about morphology. We are talking about cell level. What about gene level? We know the human, we have 20,000 protein coding genes that make us each of the cell. And at a gene level, which gene can drive the speed differences between human and mice during the development and or the stem cell differentiation. So during the early development, we know a human is slower, but the question is how many genes can contribute to the speed differences? We don't know. And for the tissue regeneration, we know the blastema differentiation can forming the structure of the limb in axolotl. And the mouse is an amputation level specific regeneration. Specific means if you cut the tip, it still can regenerate you partial of the tip. And the question is, why not the whole process of the mouse tissue regeneration versus axolotl blastema differentiation? Is there any similarities between two cells or not? So we don't know yet. So the biological question can translate it to the statistical question. That is, if we have two, paired the data. Let's say if we have human or have the mouse, because we have 20,000 protein coding genes, and each gene we can perform the paired study. Let's say similarity and the differences between the human and the mouse. So specifically, the biological question were translated to basic statistical question, that is how to identify gene pairs that exhibit with similar temporal patterns, and how to quantify the rate or temporal discrepancy between the two temporal trends, especially if they share similar patterns but vary in their pace or just a shift. Basically means the statistical question is how we can doing the temporal alignment or comparative time course data with two different species or one species in two different conditions. So for the comparative time course data, there are multiple confounding factors making this very complicated. First thing is the pattern complexity. This is just an example. Let's say the pattern can represent gene expression changing over time. This can be the human versus mouse, or in my point is the human versus mouse during the early development, or the mouse versus axolotl during the limb regeneration. So basically, there are just two conditions, but we are looking at a specific gene expression level, not a morphology. So the pattern complexity is the compounding factor that makes the whole comparative study very challenging, because we cannot doing same-day comparison. And data uncertainty means when you're doing time course data, the first thing you're thinking about is to sampling the data. We are sampling at an hour, 1 day, or 1 month. This is a question, because depending on the pattern, the sampling density will dramatically affect the pattern. In the context of biology, because doing biology is apparently very challenging. It's not straightforward. It's not like a record, a sound, or video. So basically, at each time point we're spend a lot of time. So giving the certain lab space or the certain budget, so how many samples we need to generate a time course data is a question. The third confounding factor is data noise. As you know, we have individual variations, we have technical variations, so all these kind of variation can make these things more complicated, especially for time course data, because time course data essentially the question to address the pattern similarity and the differences. Given such variation, how you can subtract the useful information from the very noisy and low sampling density data is a question. So in this study, I will introduce our package called time meter. So basically, I developed a time meter. Time meter is a dynamic time warping based statistics framework to assess temporal pattern similarity and the progression differences. So you can check the time meter in GitHub, my GitHub. And the paper published in NAR a couple of years ago. So what problem to solve for Tom meter? We solve two major problems. The first problem we want to solve is the pattern similarity or not. So basically, Tom meter is trying to assess the temporal similarity and the differences. The second question the Tom meter want to address is the progression differences. So if you have two Tom core data, pattern very similar, but the progression are different, either during the shifting or speed of change are different. And how to quantify the differences is the question we want to address in the Tom meter R package. So what are the statistical problems? When we talk, so basically the statistical problem will break down to multiple steps. The first thing is how to align the Tom course data. If you have two Tom course data, how to align them. Align means we project the day one in one condition to maybe day 2 or day 10 in another condition. In the human, let's say, in day 5, equivalent to mouse, day X or day Y. So basically, how we can align the temporal patterns based on statistical framework. The second question to address is how to measuring the pattern similarity after we align them. So basically, the pattern is complicated. When you have different progressions, they'll be very messy. How we can trim and cut and doing to quantify the similarity of two patterns. So if you can see this, it's just a shifting. So, and this one, they started at the same time, but have different speed. So how to quantify the process is a question we want to address. So Tom meter using a dynamic time warping algorithm to as a step number 1 to align the two Tom course data. So dynamic time warping algorithm is to calculate the path that can minimize the distance of the two temporal sequence. So you can see that there are two Tom course data. So basically, the question we want to address is how to align them to make the equivalent. Let's say day 1 equivalent to day X, day Y. So the DTW algorithm essentially trying to put the data into a 2-dimension, then find a path that can minimize the distance between two. So basically, based on this, actually DTW is widely used in speech recognition to understanding how to warping the Tom course data and make them, comparable. So this is step number 1. So step number 2 is how to trim and truncate the pattern to maximize the post-alignment rank correlation. So basically, when you have Tom course data, we can project in a way that day 1 equal day 2, and day 2 equal day 5. And then we can calculate experiment rank correlation between the two Tom course data. But there are many places can align very messy. So how to truncate them that can maximize the correlation is a question we want to address inside the Tom meter package. After the first 2 steps, the last step is how to evaluate the results. So basically, when we align the Tom core data, when we truncate them, and what is the measurement can be used to define similarity and differences. So basically, what is the measurement that can join certain characteristics of the two Tom course data? So I break down the question into 4 measurements. That is percentage of alignment of a query and percentage alignment of the reference. That is when you're using the DTW algorithm to align to Tom course data as a truncation. Let's say this is Tom shifting example. So after alignment, we truncate in a way that makes them the most similar because the black line actually start earlier than the red one. So because we are looking at the same time window, essentially certain part of the progression of the black line should be truncated, make the two patterns similar to maximize the similarity. Then because this one truncated partially, the percentage alignment is calculated by the untruncated divided by the whole term course. So basically, the percentage alignment represent what a fraction of the data can be similar to another. So basically, this is a measurement. So what we want is we truncate less and maximize the similarity. The pattern similarity is calculated by simply experiment correlation. You have the paired the data. And the p-value is calculated by the shuffling. So basically, we repeat the process many 1,000 times, just shuffling the data and then calculate everything, like the pattern similarity, to asking the likelihood of by random chance, the pattern similarity is greater than the original data. So the p-value is calculated by the shuffling. And here is this is the similarity data. So basically, let's say, if you see the two lines, they are different speed. So speeds are changing. So basically, they are changing over time. And when we're doing the temporal alignment, dynamic time warping, project to align the time, we can simply project the data into the time of query and time of reference, representing which time point can be equivalent. So if you can see, when the orange line changing the speed and quicker and quicker, the slope actually increase. So intuitively, the slope of the line indicated the speed differences, the full chain speed difference between the two. And if we have a time shift data, let's see the speed are the same, but just a delay. And this were represented as the shifting of the line when we after post the dynamic warping alignment. So two characteristics actually allows us to visualize and understanding the pattern, just project in additional space. And then we can define a PAS score that's called progression advanced score. So advanced progression score is defined by the average deviation warping from the diagonal line. So intuitively, if the two time course data are the same, the time point should be follow the diagnosis line. So the more deviation from the line indicated there are a lot of differences. So if you see this case, the orange line, the speed always changing, and they are shifting to high speed. So slope are changing. Essentially, the area will be getting bigger and bigger. And for time shift data, the speed never change. However, depending on how many days, how many hours you delayed, the progression have a systematically shifting. So basically, time shift is special case, PS4, equivalent to shifting time. And if the patterns, let's see, the patterns contains multiple sub-patterns. And for example, the query contains two sub-patterns. If you see the time days, the first 50 days, the red one is much faster than the black one. But after the 50 days, the speed are the same. However, because the first 50 days, there is a speed differences generate a shifting. So this is called a mixture pattern. And how the time meter dealing with mixture pattern. So basically, after the dynamic warping, algorithm and trimming and something like that, and the data will project to the time-to-time space, and the different speed of shifting will be represented or characterized by different segments. So basically, when you have the speed differences, the slope were showing the difference. So basically, the time course data will be broken into multiple pieces, then using a segmental regression to fitting the curve. So basically, the segmental regression, as you know, we have to determine the breakpoint. So it's a trade-off between the number of breakpoint versus the overfitting. So there are trade-off, underfitting and overfitting. Intuitively, as you know, for the segmental regression or the machine learning models, the more parameters to add in, the better performance will be. However, we generate overfitting because each of the parameter adding will potentially cause a problem for overfitting. Then the BIC essentially intuitively generate a formula that keeping a penalty to the number of parameters to adding. So you adding one parameter, let's say in this particular case is, if we adding in one additional segment, we'll give some penalty. So the performance significantly increase will overcome the penalty we got, then we'll choose it. So basically segmented regression, the breakpoint is determined by a minimum Bayesian information criteria. And then once we determine the different segments, we can calculate the area of the differences between diagnosis line and calculate PS score. Then I was talking about a few applications. The first application we did for Tom meter is comparing the human and the mouse embryonic stem cell differentiation. So we know the human always slower than the mouse in terms of the early development for, especially for gestation period is very, very slow. We take the human 38 days, the mouse only 21 days. When we put in the dish, like in vitro, so the mouse cell differentiation always much, much quicker. So at the morphology, at the cell level, mouse, we know mouse is faster. But the question is how many genes can driven this whole process is unknown. So specifically, let's pick up gene. The gene in human day 20 equivalent to mouse day 3, 5, or 10, we don't know. So what we did is we generate data for the human embryonic stem cell differentiation into the neural lineage, and using the mouse epiblast cells to differentiation into the neuron differentiation. Then, because for the human and mouse, usually we have 18 to 20,000 protein coding genes. So each gene, we can look at the human pattern versus mouse pattern during differentiation. And then to detect which gene, protein coding genes, have a similar pattern regardless of speed between two species. So Tom meter detect 1,461 similar pattern genes. This represents 10% actually between the human and mouse in terms of the Tom course similar genes. So here are some examples. The red one are the human, cell differentiation, and the black one are the mouse. So basically they are similar regardless of the progression or delayed, whatever. So this represents 10%. And not surprisingly, when we calculate the PS score, the human is way more slower than the mouse. However, every gene shows different. So basically most, like 99% of gene human have a differentiation in terms of the temporal pattern. Gene expression is slower than the mouse. Let's see, there are some examples. So basically, this is consistent with the morphology data. However, we just the question is how many genes can driven change? The answer is 10%. Basically, 10% protein coding genes can driven the whole systematically differences between the human and mouse during cell differentiation. There are similar patterns. Of course, there are a lot of differences, but 10% of the genes show very similar pattern, what they call conserved pattern. But 99% of the similarity patterns, the mouse is way more faster than human. The second application is applying the Tom meter for the limb regeneration data to understanding the similarity between axolotl plastima cell differentiation versus mouse digit regeneration. So we know axolotl can fully regenerate a limb. So and the mouse digit is partially can regenerate. So if we cut a small piece like tip, they can regenerate. And if you cut further, they can't. So the question to address is, if we comparing the blastima for axolotl, this is essentially driven the limb regeneration. How many genes showing the similar pattern with the mouse limb regeneration? So and we generated both the axolotl blastima differentiation Tom course gene expression data and the mouse digit RNA sequencing Tom course data, then compare together. So here are examples. We declared many. So basically, there are many genes, like say PDGF is the growth factor. The red line is not digit regeneration. We collect the wound bed, then doing sequencing, this pattern. So it's real time, the day, hours. And the black one is axolotl-blastima differentiation. It's from a different data set. So basically, there are many genes showing very similar patterns between the axolotl-blastima differentiation versus mouse limb regeneration. And then we put them together to ask the question, what is functional relevant? Then we perform the pathway enrichment analysis to understanding the biological functional relevant. So basically, they are top enriched pathways. It's mostly related to growth factor and telomere and wound healing. So this makes a lot of sense. So basically, they are the limb regeneration. So yeah, I mean, this is so thank you for. I was from the Thomerson and Ron's lab. So basically, I was a computational biologist just in Morgridge at Morgridge Institute for research. And also, there are two major collaborators, Connie and Ray, and for now digital regeneration. And our research funded by, the DARPA and NIH. And also, this is my lab. Currently, I have one postdoc, two students. So yeah, that's it. Any questions?
It's fun.
Yeah. Hello?
Hey, can you hear us okay?
Oh, yeah, I can hear you, yeah.
Yeah, I have a question for you. I'm curious about the details of how you work with population data, because I understand how this works with maybe one mouse and one human. Yeah. But how do you deal with population data? Do you sort of concatenate the time series and then perform the time meteor analysis, or how does that work?
So for the application, we just calculate average. But actually, there is a way to do this. So basically, either we calculate the average. If there are paired data, we can calculate the post alignment and then calculate average eventually. So basically, you can calculate average at the beginning before the warping. Or if there are paired data, let's say the iron sequencing data, transcription data, or the protein data, you can calculate the alignment and then pulling together later on. So there are many ways. So currently, all the application in my paper is calculate the average. Before calculate the alignment.
I see. Thank you. That makes sense. Yeah. And then another question for you. I know you did this work back in 2020, I think. Now that transformer models are everywhere, would you consider using a transformer model for something like this, or why not? Yeah,
I agree. We are thinking about that.
Yeah, gotcha. Well, to me, I actually have a hard time conceptualizing how you would set this up as a transformer model problem, because you need a ground truth still, and it seems like you don't have the ground truth here. Maybe there's a way to do it. I'm not sure.
Yeah. So actually, because of time, I don't have time to showing the most of study in the paper. The one major problem is there is no ground of truth for evaluate time course data. The only way to do it is to validation. However, it's challenging at the time, especially when we're doing the data science. We have to convince our collaborator doing tons of work. The way I address the question is based upon comparing its real biology. So basically, there was a published paper talking about the consensus between the human and the mouse. It got like many genes are similar. And I run the same data set and then put a Venn diagram. What surprised me is the time meter detect a subset of the gene that the paper claimed. So, and then I'm trying to figure out the differences. So basically, the major conclusion in that paper is certain pathways are enriched for the whole group. And then I punch out the one overlapping with time meter, all the GO terms, not significance, that I put in their significance. Then I calculate separately. So basically, the whole pattern is driven by a subset of genes, but not all. So indicating that the time meter is more specific. So this basically is the most specific. This is one way to demonstrate this. The second way to demonstrate our methods is good is we look at the data manually. So basically, to see the pattern, if we see the pattern is similar, we call it significant p-value is great, and then compare with other methods if they caught nothing recent, there is a problem. So time course data always have a lot of challenging for validate them, but we can figure out indirectly trying to validate our methods. This is the way I did in my prior study.
Awesome. Well, I'd love to ask you more questions. Unfortunately, we have to stop here for time, but let's thank Peng one more time.
Yeah, thank you.
Give me a moment to resituate our screens.
Okay. Thanks for having me here. I'm going to talk about our recent work on controlled differential equations on long sequences via non-standard wavelets. This is joint work with Jiang, Peng, Satya, and Vikas. So effectively capturing long sequences is one of the challenges in sequential modeling of data. These data arise very oftenly in language models as well as multivariate time series data from physiological data pertaining to, let's say, heart rate, ECG, EEG, etc. A large number of models have been already developed for these purposes, ranging from RNNs, LSTMs, and transformers. However, today we will look at a different approach. Insights from differential equations have been brought together along with the powerful representation capabilities of neural networks. For example, in neural ordinary differential equation, data is observed at a sequence of time points. And on top of that, we want to model some hidden state C. The move from ordinary to controlled differential equations is that now the hidden states can be dependent on a continuous path X, which indeed depends on the data that was observed at these different time points. So you can see that the hidden state for ODEs and CDEs, there is a difference in the fact that there is a continuous dependence on a path, which can help us modulate the hidden state better. This is actually one of the key differences between other models such as LSTM and RNNs, which take a more discrete stepping in time. There has been a lot of work involving ordinary differential equations. However, this work focuses on controlled differential equations, which have been proven to be a generalization of ordinary differential equations. So let's have some motivation for why do we need to go to controlled differential equations in the first place. A drawback of sequential modeling using ODEs is that the solution to an ODE is dependent on the initial state, and there is no way at later points in the time the trajectory can be modified. This is out of scope, and this is problematic in several scenarios. For example, let's say we are interested in measuring the heart rate or oxygen saturation level of someone. These kind of depend on what activity the person is performing. You would expect them to vary while you are running differently compared to while you are maybe just sitting down. This is just one example. Even in case of fMRI data, the activation levels on the brain are dependent on the functional activity we perform. So this kind of data arises very often. And ODEs, because of the fact that they are kind of tied to this initial point, do not have the ability to being controlled or modeled by modifications that happen to the trajectory later on in time. And this is where the controlled differential equation via the control path comes in. So now that we have seen why do we need controlled differential equations, let's see what is the difference between the ODE setup and the CDE setup in terms of the formulation. So you can see that it's kind of pretty much the same equation. The only difference is that now the integration is not a Riemann integral, it's a Riemann-Stiles integral. And in most cases, what is assumed is that the path X is kind of continuous. So we kind of replace it, we take out it and replace it using the derivative of the path. So our solution to modeling long sequences using CDs involves the use of an integral transform. So what we do is we take the CD and cast it into an integral transform via transformations. The details of which are there in the paper, but I will skip these in the interest of time. But these are, at a very high level, these are just projections from one space to the another to get us to a stage where we have a kernel which corresponds to the integral transform being performed. So the question is like, well, now with that we have this kernel view of the integral transform, how do we solve it? One approach is to discretize it. So when we discretize this equation, we end up in a simple matrix vector form without all of the. So ordinary differential equation solvers are well known. People have been using this, but the solution approach in our case is solver free. So we just use discretization to go from the integral transform to a matrix vector product. All of this is good unless and until you want to look into long sequences. And you would imagine if the sequence length is really long, these matrix vector products are like prohibitive because of the squared complexity. So this is where the multi-resolution analysis comes in as a tool to help us deal with these large matrix vector products. So essentially we use wavelets at different levels and different translations. So the easiest way to think of these would be feature extractors giving us different resolutions of the data that we are observing. And these are basically implemented using convolutions. The filters are kind of predefined here. So let's look at how the decomposition of this. So this is the kernel matrix that we will get out of the small a. And on top of this, if we perform the 2D multi-resolution decomposition, we will end up with like 4 different component matrices. And each of these are essentially 2D integrals involving the wavelet functions. So we'll have alpha, beta, gamma, and then the A matrix at the next level. So the double integrals can also be represented in terms of matrix vector products, which is has been laid down here. So still we don't see a computational benefit so far. This is where we need to use some special properties of the operator that we are working with. This is a result from like 2 decades back from Belkin, Koefman, and Rochlin. And they showed that for kernels of the class of Clarendon-Zygmunt operators, they have some very nice properties, which is like they're bounded away from the diagonal. And not only the function, but also the derivatives of that function are bounded. What this leads to is the fact that the 3 component matrices that we had previously seen will be diagonally banded. And this is where the sparsity comes in and makes computation feasible for the large matrices. So let's look at an example. So this is like the full-blown matrix. So we have taken a lower triangular form because for the applications that we considered, we don't want our prediction at a current time point to be dependent on future time points. So that's why the lower triangular part is there. But this is, so there is no sparsity as such here. But as soon as you hit it with the 2D non-standard decomposition, you will start to see that we start getting a diagonally banded structure. I don't know if it's visible, but you can see that everything else is 0 except from very close to the diagonal. So this is where the sparsity comes in. So if you keep doing it for more and more levels, you will see this structure even more clearer. And so the number of levels is kind of a hyperparameter choice that we have. And at some point where the matrix vector product is feasible, we just stop at that point. And rest everything is diagonally banded. So these can be instantiated pretty easily. So if we write these different steps together, it turns out that there is a recursive formula to this. but there are still things that need to be tackled. So far we have seen that we will get a diagonally banded structure, but from empirical evidence, this is heavily over parameterized, and like it's very hard to generalize using the data that we are interested in. And this is primarily because, so if you think of these diagonally banded things being convolutions, so you can see that the weights are not being shared, so they are varying all across the length. So as a regularizer and a kind of a simple choice, but effective one, what we do is we limit the sharing. So we introduce some sharing in the parameters, but also do unsharing. So that's why we kind of refer to as this partially unshared convolution. So you can see the different colors here. So these are kind of shared parameters, but at some point they are also being unshared. Whereas on the right-hand side, everything was unshared. So this is very hard to regularize it. So this is kind of a regularization which makes learning possible on the data that we are interested in. And because we do this at all levels, and as mentioned previously, the dense matrix only needs to be materialized at the coarsest level, and that is not a problem. So here is kind of the full pipeline. So we start with some control path. We perform like wavelet decompositions to get details and approximations, coefficients at different levels. These are then transformed using the parameterization that we have seen. And finally, we use the inverse transform to get back the response path. The benefit of using wavelets as kind of the tool that helps us go to the latent space is the fact that there is a natural inverse transform and a fast one that's associated with it. So coming to the evaluation of the model, we check it on 3 different kind of tasks. One is like standard benchmark problems, which are like regression and classification tasks. And then on a sequence to sequence modeling for long sequences. And lastly, we also come across this nice body of like coupled differential equations. So in standard benchmarks, so this is like a data set called BIDMC32. So the task is to regress to respiratory rate, heart rate, and oxygen saturation level. And the input is ECG and PPG sequences of length 4000. You can see that BCRD, which is our proposed framework, works reasonably well to compare to the rough differential equation, which is one of the baseline models. And in many cases, it is like orders of magnitude faster than the existing baselines. Similarly, for classification, here the sequence length is longer. So this is like 17,000. And the task here is to classify eigenworms into 5 different classes based on their motion pattern. Again, you can see the proposed framework performs comparable to the existing baselines, but in much lower time. So the noise variant is one of the variants that we tried. It basically tries to perturb the coefficients in the latent space, and we see that it can give us some benefit in terms of generalization. So for sequence to sequence modeling, we tried the standard autoencoding, denoising autoencoding, and masked autoencoding. And in all cases, you can see that the time taken by BCRD is like much less. And the performance is also, so except for the NCDE, the autoencoding task is kind of simple here. So it doesn't really show which method is powerful. But with denoising and masked autoencoding, you can see the difference clearly. So lastly, we look into coupled differential equations. So in many dynamical systems, there are there is one independent variable, which is like mostly the time, and there are multiple dependent variables, but these have like couplings in between them. So one is dependent on the another, not just time. And because of the fact that BCRD models the system as a integral operator, so you can imagine that it helps us to transform from one function to the other. And so we test this on like several well-known coupled setups, and except for the toy setting, you can see that BCRD will give you the least MSE, also in the least possible amount of time. So this clearly shows that there is value in modeling the dynamical system as a integral transform in settings where we have coupling involved. Here are some plots. So again, so like you can see for the Lotka-Volterra system, the prediction of our model is like as close it could get to the true one, whereas for the baseline models in all the three cases, it's quite different. Similarly, for like the chaotic Lorentz system, BCRD gives almost perfect reconstruction, whereas the CDE setup is not able to do so. So in conclusion, we present BCRD to model control differential equations via an integral transform and multi-resolution analysis. The strategy is to model long sequences but fixed length by unrolling the dynamics, which is like discretization of the transform to get a matrix vector product. Empirically, we observe that it is best when the number of levels is large, leading to a small but effective coarse representation of the signal. And BCRD provides an efficient way to handle coupled setups. Our code is available on GitHub, and please reach out with questions or comments. Thank you. Happy to take questions at this point.
Sourav. I want to get us started. So, as I understand it, these ordinary ODE models, one of their primary benefits is that it allows you to model continuous data, so you don't have to discretize. I think I got lost during the math because at some point you do have to discretize the model. Do you end up losing that continuous?
Right. So the approach that we take is closer to discrete, but we start from the continuous form and we discretize at the point where we just need to solve the matrix vector product. So other cases, the stepping would be done through solvers. So you can sample it very finely. And if you sample very finely, this is where you will get to the long sequences. And so that's where the bridge is between the continuous to discrete in our case. Yeah.
And if there's no other questions, how should we be thinking about the number of parameters of these models?
Oh, these are actually like very lightweight. So you could see from the training time, these can be, so for the coupled setup, these can be trained in like less than a minute. So I think these are almost always less than 100,000 parameters, except for the regression and classification tasks. So those are like 100,000 max. The other cases we can do with much less actually. Yeah.
And sorry, are you using a neural network to actually?
Yeah. So the parameterization that you saw in between for the diagonally banded matrices, these are through neural networks.
Sorry to you, but are there you give any examples where maybe continuous data is needed to do a modeling effectively?
So I think like mostly these data coming from physiological setups like heart rate, respiratory rate, ECG, these kind of things.
Right.
These are what people have been looking using these DeFiQ setups.
You could just discretize that data and use the high sample rate, right?
Yeah. So again, you will get to longer sequences, and long memory across long sequences is one of the challenges. So.
So when transformers start to fall apart.
Yeah.
Any other questions? All right. Let's thank our speaker one more time.